Welcoming you all. This is Adil Maman. The topic I am going to present is flagella, bacterial flagella. Flagella is composed of subunits of a low molecular weight protein called as flagellin, having 20 to 40 kilodalton. It is hair-like structure, microscopic, involved in the locomotion, as well as it is considered as motile organelle composed of thousands of protein subunits. Structure: the flagella is a helical structure composed of flagellin protein. The flagella structure that is divided into the three parts. Here you can see the GIF representation of those parts: basal body, hook, and the filament. Basal body. Basal body, it is a center rod inserted into the series of rings which is attached to cytoplasmic membrane and the cell wall. These series includes L ring, P ring, MS ring, and the C ring. L ring, it is the outer ring present only in gram-negative bacteria. P ring, it is a second ring anchored in peptoglycan layer. MS ring, that is anchored in cytoplasmic membrane, and the C ring, it is anchored in cytoplasm. Here you can see the image showing us the various parts in the series of rings. Here you can see that L ring, it is an outer ring anchored in the lipopolysaccharide layer and found in gram-negative bacteria. P ring, that is anchored in the peptoglycan layer, and the C ring anchored in the cytoplasm, and the MS ring, that is anchored in the cytoplasmic membrane. Second part, that is hook. It is wider region at the base of the filament. It connects the filament to the motor protein in the base. The length of hook is longer in gram-positive bacteria than the gram-negative bacteria. If we can further describe this, we can say that hook is a curved structure that connects the basal body to the filament that originates from the cell surface, and the hook acts as a universal joint that connects the flagellar base body and the filament. After hook, we have a filament. The flagellar filament is composed of single protein called as the flagellin. Its hollow structure originates from the hook. The filament is what it is supercoiled. If we talk about the types of the flagella on the basis of arrangement, we have monotrichous, amphitrichous, lophotrichous, and word peritrichous. Let's talk about monotrichous. Presence of single flagella in one end of a cell. We have examples Vibrio cholera, Pseudomonas aeruginosa. Number second is lophotrichous. That is the presence of bundle of flagella in one end of a cell. Example Pseudomonas fluorescens. Number third is amphitrichous. Presence of single or cluster of flagella at both ends of the cell. We have example aquaspirillum. Number fourth is peritrichous. Presence of flagella all over the cell surface. Example E. coli, Salmonella, Klebsiella. Atricus, that is absent of flagella. The functions of the flagella: they help an organism in movement or locomotion or motility of an organism. They act as sensory organs to detect temperature and pH changes. Few eukaryotes use flagellum to increase reproduction rates. Recent researches have proved that flagella are also used as a secretory organelle, for example, Chlamydomonas.